Welcome to our soil science composting webinar with the Master Composter Recyclers, Dr. CeCe Crosby.
I can tell you a little bit about soil structure and organic matter. And then my specialty is microbes. I'm a soil microbiologist by trade. I did a whole bunch of work looking at microbes in compost and compost tea, and how do those communities look and how do they change. So if you've got questions about soil microbes, toss them out there. And if I don't have an answer, I can probably help you find one. And then the other thing we like to talk about is what's actually happening in your compost pile. So we'll take a look at some compost chemistry and look at how we take plant material and turn it into those long-term carbon molecules that we keep talking about for garden health. You'll notice on this graphic that I've pulled up kind of a zoomed in view of what's happening in a soil. And so we've got our sand grain, we've got some clay particles. Silt's not represented on here, but you can imagine the silt as being halfway between those two in size. And what you'll notice is the scale of the root hair as well. And then you can also see some organic matter up here. The piece I like to draw our attention to is if you look at the clay particles in chat, what are you noticing on the surfaces of the clay particles as opposed to what you're seeing on the surface of the sand? There's a really distinct thing happening on the clay. Yep, so the soil texture is critical with the clay. You'll also notice that the clay has all those little white minus signs on it, right? Yeah, so we've got charges from cations on there. So the clay itself actually has a whole bunch of little teeny tiny negative charges built into its structure. So if you were to get in there and look at the crystalline structure of the clay, it has that charge physically built in. And the only way that it can neutralize that charge is by picking up positively charged ions from the water around it. So things like calcium, magnesium, sodium, ammonia, those are ammonium, to be more specific. All of those are going to stick to clay particles. So when we're talking about soil nutrition, we're actually talking about what's stuck to the clay. So yes, clay is really important for how water moves, but it's also really important for physically holding your nutrients in place. In soil science, we call it cation exchange, which basically means that your clay is going to hold cations and your roots are going to trade with the clay for those cations. So they're going to take ions that they don't need, usually hydrogen, and they're going to trade with the clay to get things like calcium and magnesium and certain forms of nitrogen and pick those up themselves. So clay is your nutrient bank. You'll notice the same little negative charges on the organic matter. And even more importantly, some forms of organic matter can have positive charges too. And where you have those positive charges on your organic matter, it can hold things like nitrate or phosphorus or sulfur that are negative ions. So having organic matter in there is actually going to give you that advantage. So when we look at texture, we often only think about what's happening with water and that the sand grains make bigger pores, so more water goes through. But we also want to think about nutrients because it's the clay pieces are really important for that. With your shaker test, essentially what you're going to do is you're using water to separate all those soil particles. So by adding an excess of water, we get the organic matter, the sand, the silt, and the clay to separate from each other. And when you shake it really vigorously, essentially what you're doing is you're dissolving the organic matter in the water. So you may notice that your water has kind of a dark color to it now. The sand settles out immediately. You almost can't keep it mixed in with the water. The silt will settle slowly over 10 or 15 minutes. And the clay might take overnight or even a week to fully settle. And some of your clay may never fully settle. So the clay takes a while. In fact, when we do this in a soils lab, we actually put these in little conical tubes and we spin them really, really fast in a centrifuge to get the clay to come down. And then we can get a really careful measurement on exactly how much of each one that we have. But in the shaker jar, you get a pretty good estimate. But you're going to determine how much of your total soil solids are sand, how much is silt, and how much is clay. And then on the three sides of this triangle, you've got numbers for each one. So along the bottom, you have your percent sand. So say you have, 15, or let's do 10. 10 is a good number. So say you have, a total of 10 centimeters of material. If 4 centimeters of that is clay, is sand, that would give you 40% sand out of your 10 centimeters. So you would use this horizontal line right here. You would follow this 40% at the bottom, and you'd follow the line going from that up. And then you can measure your silt. And if your silt is 2 centimeters out of the 10, then you would start at the 20% line there and follow the horizontal line from that. And then finally, you don't necessarily have to measure your clay, although since we didn't know exactly how deep the original material was, you probably will want to. Your clay layer is usually the smallest, but in this case, I have 60, so I'd have to have about 40% clay left. So I would follow my 40, 40 and 20, and that should lead me, double check that here, that should put me in the clay or clay loam category, depending on exactly how I follow that. So I can determine my category by tracing all three of those. So in this one, you can see sort of where the three layers are. We get our best estimate. So you measure the height of your layer. You can use whatever unit you want, so long as it's the same unit for any of these. So if you have an inch ruler, get an estimate in inches. If you have a centimeter ruler, that's a little bit better. You could use your centimeter ruler and get an estimate in centimeters. And like I said, on this one, if you noticed on yours that your clay didn't settle out, put the material that you want to test into your jar and kind of give it a little pack down, measure it, maybe mark it on your jar so you don't forget, and then mix in your water and shake it, because then you can just subtract the sand and the silt and you know how much clay is roughly left in the water, because it would be essentially the missing volume would be clay. So
question.
Yeah, absolutely.
So what's the best reading to get if you want to grow food?
Things that say loam are generally kind of the coveted soils. So the loamier something is, that means that it has sufficient aeration, but also good nutrient holding capacity. So if you have something that's a heavy clay, so if you're in the clay category, you're going to have a really hard time getting water to infiltrate into your roots. And you may have places where your roots will have a hard time getting through. You can't really change your soil texture on a large scale. So often we'll have people, pot or put their plants in with a plug of compost or, a plug of like peat moss or something to give them some airspace. And I mean, your garden texture will improve with time if you keep adding organic material. If you have something like a sand, you're going to have really low nutrient capacity, but really great filtration. So you might, also be adding organic matter to hold some of that water in there, or you may have to add water and nutrients more often to get your food to grow. So, I mean, it's great to have a loam, but at the end of the day, your soil texture is what your soil texture is. So you have to work with it. So, there's a lot of information you can get about how to manage your soil with the texture, but at the end of the day, the piece that it gives you is sort of, it's not necessarily something you get to shift, right? You can work with it, but the land that you're working is going to be that texture no matter what you do. That's why a lot of people do the above ground gardening. All right, should we take a peek at what's happening inside some of that organic matter? One of the things we like to talk about, and this is one of the coolest parts of being a soil scientist and of playing with compost, is that when we look at soil, it's probably one of the most diverse habitats on the planet, but it's also one that we just barely understand. I spent almost 10 years of my life studying soil science in a research lab and a school setting. And the people that are at the top of this field look at it and they say, oh, we know less than 10% of the organisms that live in the soil. We know they're there. We can find their signatures. We can see their activity, but we can't necessarily culture them or define them or give you a good picture of exactly what's present. It's just such a complex and kind of amazing habitat. So we're going to talk about what role bacteria and fungi play in actually taking the materials you put in your compost bin and turning them into that dark humus-rich material that we want in our garden. And then we're also going to talk about what benefits those have in your soils around your plant roots. So why do we want those microbes in our habitat in the first place? Because they're a very important piece. So if you look at a breakdown, and there's a lot of numbers here, so let me break some of these down for you. I'm a nerd. I love numbers. So imagine for a second that we went out and we looked at an entire hectare, so basically an acre of soil. What you would find is that in that hectare, we've got about 400 to 5,000 kilograms of bacteria. And to put that into imperial system, sorry, a kilogram is about 2.5 pounds. So you're looking at like 800 to 10,000 pounds of bacteria just in that top layer of soil over about an acre's worth of land. So kind of to put that into perspective, a cow is about 1,000 pounds. So you're looking at 10 cows worth of bacteria, and that's not even looking at the fungi, the actinomycetes, or the algae that are present. So you'll notice fungi is about 10 times as much. So huge numbers of organisms are living in a hectare of soil. So yeah, you got about 15 cows worth of microbes in a hectare of soil. You've got about 1 billion bacterial cells in a teaspoon of soil. How do we know that? Well, we can look at that. So I can make a microscope slide smear of that soil and detect the bacterial cells. And we can also detect their DNA if we extract that from the soil and take a look at it. So we know they're there, but they don't really like to grow. So once we take them out of the soil, we really only see about 1 to 10% of the organisms that we think are present. Pretty crazy when you think about it. So there's more. You're looking mostly at the bacteria here, but we have an entire ecosystem happening in just our soil that's centered around the organic matter that's in your soil. So down at the bottom of the screen, in the bottom left corner, there's essentially your compost pile that's labeled organic residues. So that's things like leaf fall and old plant roots and the bodies of all of these organisms when they reach the end of their own life cycle. And everything else in your soil is going to be feeding on that. So this is what we call our primary layer. And the organisms that feed directly on this are what we call primary producers. And they're going to be taking this material and turning it into their own bodies. These are things like bacteria, fungi, actinomycetes. Some earthworms are going to be eating that material directly. And then from there, these all have predators that prey on those. So we've got mites, we've got springtails, we've got larvae, we've got protozoans, which is one of the things that popped up in chat as something you guys are really interested in. So protozoans are single-celled organisms that sometimes 2 or 3 cells or little communities, but they're bigger and they're more complex. Those guys are going to feed on the bacteria, the fungi, and the actinomycetes. And from there, we can get another layer. So you can get 2 or 3 layers of microscopic communities that are all feeding on each other and back on this organic material. So the organic material in your soil is supporting a huge group of these different organisms. And why do we care about that? Well, like in all things that we do, diversity is a great thing, right? Not just within our societies and within, within the animal kingdom and the plant kingdom, but also within our soil kingdom. The more diverse your soil biota is, the more likely you are to not have plant pathogens that are getting supported in that soil. You're going to have more cycling of nutrients. You're going to have more nutrients available. So a healthy soil is going to have a great diversity of microorganisms and of, I don't want to say macroorganisms, but kind of that mid-level as well. So all of these are going to be really small. I mean, this little beetle here is 20 millimeters. That's visible, but it's pretty tiny. A set or two set, your thumb is about 2 centimeters or so. So if you look at just the first knuckle of your thumb, that's about 2 centimeters. So that's about the size of some of these bigger ones. So if you think about 50 millimeters of a centipede, that's just a little bit longer than your thumb. Everything else is going to be smaller than that. So these are very, very small organisms. When we look at composting, we're also looking at a community. And all of those organisms that we just looked at in the soil food web are going to be present at different times in your compost. Yeah, if you're doing one-on-one bins, so if you've got something running at your house, you can think about what it is that you're putting in to your initial pile, right? You're going to have food scraps. You're going to have coffee grounds. You might have different types of yard wastes. You might have leaves or grass clippings or even newspaper or cardboard or other organic household waste going into your pile. And those are all the easy foods, right? So in your first stage, when you first get that pile together, what's happening is bacteria are having a party. You've basically set out a bacterial buffet. They're going to come in, they're going to grab all of those easy food sources. They want sugars, they want proteins, they want fats, they want food sources that are very, very simple to break down. And they're going to get that out of all of those feedstocks. And as they break those down and metabolize them, their bodies are going to produce heat. And those more difficult to break down substances like the cardboard and the leaves, and if you've got wood chips in there or straw, any of those kind of brown materials, those don't break down as fast, but they do give a lot of aeration and a lot of insulation. So as those bacteria eat the easy food, they generate heat from their metabolism. The browns insulate, your pile gets hot. And that first round of bacteria says, oh, I don't deal with high temperatures. I'm going to get out. So that first round of organisms heats the pile, and they basically make their own house unlivable. It's too hot, they get out. The next round comes in, and we're going to get what we call the thermophiles. Those are also bacteria, and they exist at temperatures over, now I have to convert in my head. I was going to say 40 degrees C generally. So you're looking at around 100 to 135 in Fahrenheit. Those organisms are going to thrive at that temperature. And not only are they going to use easy foods, but they're going to start to get into breaking down some of those harder foods, or at least softening up those harder foods. And when they've used everything they can use, the pile will cool down because their metabolism will slow down. When that happens, the actinomycetes and the fungi move in. Those need cooler temperatures, and they can eat things that nothing else can. So fungi can come in and they can break down wood material. They can break down straw. They can break down, the paper in the shredded newspapers, and they can actually get nutrition out of that. And as they do that, they transform the rest of that material from something recognizable into completely new compounds. Essentially, as we've got these microbes in the soil or in our compost, they're working on a set of really important tasks. And once we get them we got our compost into the soil, we're inoculating our soil with that. But these organisms are growing around the particles of soil. They're binding them together. They're decaying organic material and transforming it. They're helping us to release the nutrients that are contained in that organic matter into a plant available form. so that your plants can use them. They're going to help us to bring nitrogen into the system. Some of them are actually able to degrade things like herbicides or pesticides or other soil contaminants that might be present. And they're also going to help us with our soil structure. So they help keep our particles in place, to help keep them spaced, and they help us build in some airspace. So they're a really important piece. Here's what's happening. So our microbes are going to release their enzymes into the soil. So they're spitting their digestive juices into the soil. Those touch the organic matter, they break it down, they release the nutrients, and then the microbe is going to pick those up. Once the microbe picks those up, it incorporates it into its body. And slowly, as those microbes reach the end of their life cycle, they're going to decay, and they release those or those materials back in a plant available form. So one of the things that we sometimes see happening when we add organic matter to our soil is sometimes we'll see that the microbes that we add with that organic matter actually temporarily suck nutrients out of our soil. Those microbes are going to grow, and as they complete their life cycle, they're going to release those nitrogen or those nutrients again, and you're going to see kind of these pulses in your soil. So as the microbes grow, nutrients get absorbed into the microbes. As the microbes die back, those nutrients get released for your plant. So if you're adding organic material as your primary fertilizer, what you're going to find is you need to kind of get the timing down so that you get the pulses when you need them. So things might need to sit in your soil for a few weeks before you start to see nutrient release. If you're looking at your soil health, you've got a lot of different places to look, right? There's some things we can test. We can test pH. You can take a sample and send it out to a lab, or I think there's even some home kits where you can get an idea of how much nitrogen, how much phosphorus, how much calcium, how much magnesium you have. So you can test for those nutrients, but that really only tells you what's present in that snapshot. We can also look at what's happening in the biology. And in that case, you're going to see nutrients being released, nutrients being stored. You might have some interactions with microbes that change the disease pressure in your soil. And we're also going to see that soil structure is a critical piece too, in terms of maintaining water, air spaces, and kind of allowing roots to grow through your soil. So the heavier your soil, the poorer roots grow. So the question that popped up as we were coming back in is a fantastic one, which was, we talked about what's happening if you've got like a really nice batch composting where you can set up, like that ideal pile and let it heat and kind of manage it, like you might do in a composting facility or in a more managed process than your home setting. So if you're doing a home compost and it's taking you a long period of time to get your cubic yard of material, these processes don't stop just because it's not heating, right? So if you've got a subcritical mass and you're just continually adding material, the bacteria are still working. They're still degrading that material. They're still generating a little bit of heat. Their metabolisms are still running, but your timeline is going to change. So instead of something taking place over a couple of months, it might take years for that material to kind of reach that end point where you've got that nice, rich, crumbly brown material. So you can also look at it as a long game, right? Sometimes people will have multiple bin systems happening where they're sort of collecting everything they need to hit that critical mass over the course of a year, and then kind of mixing it and managing it and getting it going while they're starting their next bin to collect that next cubic yard. So you can certainly set up ways to manage that if you're willing to play a longer game and you have the space to do it. But in order to have something really classified as a thermophilic compost, you've got to get it heating. The main benefit of that heating is that you really kind of change the composition from what would happen in a normal decay process to what's happening in this like really specific decay. Well, and if you think about what's happening from the microbes' point of view, like they can't move very far, right? So if you've got a nice little clump of food and insulation, so your greens in a layer and then your browns on either side, they're going to kind of eat what they can eat, but they can't get very far in that pile. So when you turn it, you want to give them some air so they can breathe and start growing again. But two, you also expose new material to them so that they can attack that new material. So by mixing it, you're helping the process. What is it that's happening? Because we get this, I hate to use the word magic because it's really unscientific and my brain doesn't like it, but we get kind of this process that's a little bit mysterious, right? We take all of these recognizable materials, they go into a bin, they get hot, we turn them, they cool, and then when they come out, you get something that's completely unrecognizable. So I like to kind of talk about what we see with that. There's really kind of three major components that you have in your compost pile. We've got living materials. We've got dead materials. So you've got your microbes, you've got your fungi, you've got your actinomycetes, you've got your food sources, what's going in, you've got plant residues, whatever goes into your pile. And then the last piece that we have is this transformation piece. So you put in a recognizable plant material, and what we get out is what we call humified organic matter. It's dark brown, it's crumbly, it has a distinctive smell, which incidentally comes from the actinomycetes. That lovely wet soil smell that you get after a rain, you're actually smelling the actinomycetes growing. Always kind of a fun thing. So those are the main components. And when you start out, we're starting out with rich food sources, right? And we know they're rich food sources because there are food sources, right? We've got sugars, carbohydrates, proteins, we've got minerals, we've got fiber. So we put in these starting materials that have very simple, very recognizable structures. If you were in my intro class, I would have dragged you guys through all of these chemical structures for these. I'm going to be nice and not do that to us. But they're easy to recognize molecules. If we looked at our starting pile, you would recognize the material that's in it. What's going to happen is our first flush of microbes are going to eat all of that easy food. They heat things up, they die off. The next round comes in, they take whatever's left over of the simple food, and they start to kind of work on the bodies of the microbes that died. And, maybe they start on that next level. And then as we get hotter and hotter, the thermophiles come in and they eat the bodies of the microbes before them, and they start on that next more complex layer. Once all the really poor food sources are left, so now you're looking at things like fibers, so cellulose, you're looking at microbial wastes that are in kind of weird configurations. The thermophiles can't finish that material, they die back. And then the fungi come in, and fungi have really weird metabolisms. They can take all these poor nutrition sources and just chip away at them over time. And as that happens, we get this really complete transformation. So even as your pile cools, you may still see some recognizable fiber materials. You may still see your browns being somewhat intact. But those are going to continue to break down with your fungi. So our early microbes only want easy food: sugar, starch, protein, mineral nutrients. And that's going to create a lot of metabolism really, really quickly. What's happening here is your pile is actually going to shrink in size. Those easy foods get released as carbon dioxide. So a little bit of CO2 is going to come off your pile. They're going to release things like nitrate and ammonia. And the microbes that are present are going to tie up all that material. They're going to scarf down that nitrogen. They're going to incorporate it into their own bodies, and they're going to hold it along with releasing that heat. Our thermophiles come in and they tackle the rest of this. They start looking at cellulose. Lignin is the substance in wood that makes it so hard. It's very difficult to break down. The heat starts the process of softening that lignin. They deal with the remains of the food sources that the previous ones couldn't consume. They consume the bodies of their predecessors. They break down all those microbe bodies. After these heating stages, you're going to see a lot less recognizable easy food. And then curing is a stage that's easy for us to ignore, right? Because we've seen what looks like this transformation. Our pile has heated, our pile has cooled. The easy stuff, the easily recognizable stuff is gone. We've got something that looks more like a compost, but there's actually still a lot of nutrition tied up in those microbial bodies and tied up in those browns. So fungi are going to come in, and they're what are really kind of critical to a nice finished compost. They're going to work on those hard foods. They're going to work on those microbial bodies, and they're going to create a material that is more of what we would call a humus. These guys do weird things. They make weird bonds. They break weird bonds, and they make what we call an impossible or a nearly impossible food. Nothing wants this stuff. You'll see down here in this bottom corner, there's a ridiculously complicated molecular structure. And this is what fungi do with the remaining materials. They just link it together in crazy and strange ways. And it stays in the soil or in the compost for a really long time. These are the materials that wrap around your sands and tie in your clays. That's a great question, Erin. Where did the thermophiles, actinomycetes, and fungi come from? The joke that we make in microbiology all the time is that if you build it, they will come. So we kind of talk about these as being sort of everywhere. So things like fungi and actinomycetes form long-term spore structures where they're essentially living at a low level or kind of in stasis, for lack of a better word, in the soils themselves. So as you build the environment, they're going to come in on windblown particles. They come in with the with the larger organisms that sort of invade your pile. They are going to show up. If you've built a habitat for them, they will inhabit it. So you can inoculate, but you really don't need to. They will come if your pile is built. They're out there. They're in the soil under the pile. They're in the dust that blows in the air. They're all over the place. So essentially, what you're seeing is we've got these long-term storage molecules. So our soil organic matter is just like your compost pile. We have those living organisms as part of it. We have recognizable plant and animal materials. We have potential food, so all of these partially decomposed plants and animals are potential food for organisms. And as those get transformed, they're going to get into, food and energy sources, and they're also going to end up in these humified materials that stay in the soil for a very long time. So we talk about this first level as being what we call the labile pool or the fluctuating pool. That carbon changes on a daily or weekly or monthly or yearly basis. It's constantly turning over. This next level, you're looking at decades. So these transformed materials may be in the soil, 10 years, 20 years, 100 years. These non-humic and humic substances that are greatly broken down, those may be in the soil on the order of hundreds of years or longer. So this soil carbon down here in these humic substances is a very long-term piece, and it's going to be affecting how your soils act on a larger level than those labile carbons. So this is what we want. This is that material that is the reason why people add compost to their garden. Worms are fantastic. Let's not diss the worms. Worms are fantastic for that labile pool. They are constantly turning carbon over. And even more importantly, they take materials from the top of the soil and they bring it deeper down. So worms are incredibly important for the beginnings of those turnover phases. And they're also really important for aeration in poor quality soils. So worms are going to leave channels. They're definitely an important piece. If you've got, especially the deep earthworms, they come up to the surface, they take soil or they take organic material and they bring it down those burrows and they deposit it all the way down those burrows and they help it infiltrate into the soil. So even top dressing with a compost is going to slowly work that material in. It's going to take longer, right? You're not going to see as fast of a change in your garden soil by top dressing, but it does infiltrate. And I did leave you guys a spot on the document for a wrap up. So I left you, what's your take home message? And do you have a burning question that we ran out of time for? And I'll be taking a look at those. I'll probably leave this document open for a couple hours tonight, and then I'll lock down the commenting. But I'll check those out. If questions pop up, I'll send some information out through Pete or answer them directly on that document.
That sounds great. Actually, if you can send them to me, that'd be great. I'll email them
Okay.
to everyone. With our
That works.
Okay, everyone. Well, thank you so much for tuning in. Thank you again, CeCe. Everyone have a wonderful night.